Hi everyone, good morning. It's great to be here and get the opportunity to talk to all of you. I'm going to talk about how our proteins are organized in cells spatiotemporally. And this work is, as Trey said, part of a bigger project, the Human Protein Atlas project, where I'm one of the directors for the Cell Atlas work of that. And our aim, we've been working for 13 years, and our aim is to map the human proteome using our in-house generated proteome-wide collection of antibodies. And to do this, we're mainly using large-scale immunostaining and high-resolution microscopy platforms, and everything that we do is freely available in our database, proteinatlas.org, the raw data, the annotations, everything. So if you haven't used it, I encourage you to check it out. So when we map proteins in cells, we want to work with high resolution, so we're imaging cells with confocal microscopy, oil immersion, and we have some reference markers to outline the cell. This is the nuclei. And then we have a marker for a filamentous structure, the microtubules, and a marker for a membranous structure, the ER. So these are good for computational purposes to get an idea of what the cells look like. And on top of this, we use our in-house generated antibodies to systematically map each and every human protein to see where they are. If it looks like this, we can conclude this is CD44. It's a plasma membrane protein. If the image looks like this, we can conclude that this is a cytosolic protein. This is a mitochondrial protein. This protein is found in the Golgi apparatus, in the nucleoli, in actin filaments, in focal adhesion sites, or in more rare structures such as the mitotic spindle, for example. So I'm not going to show you all our images. We have been doing this for 10 years, and we have 1 million images like this that all have been annotated in terms of the protein distribution. And these images are the basis for our Cell Atlas. So about a year ago, we published a paper called a subcellular map of the human proteome, where we localized 12,000 proteins, which is more or less what you can find in a cultivated cell line, to 32 different cellular structures. And half of these proteins were previously not localized experimentally. And we could, in this work, define 14 organelle proteomes that can be used as a starting point for further in-depth studies of organelles and proteins and cellular function. And I thought that in my limited time today, I will highlight some of the interesting findings from this study and show you what we're working on currently. One thing that I find very fascinating is that a lot of images looks like this. So this protein is found in, let me see, where's the pointer? In contrast to the images that I showed you before, this protein is found in the nucleus and on the plasma membrane. This protein is found in the nucleoli and in the mitochondria. And we could, for the first time, demonstrate that as much as 50% of all human proteins are localized to multiple compartments within the cell. And we could also reveal distinct patterns of multilocalization across the cell, somewhere which this image show. I'm not going to take you through it now. But some of these were expected, a lot of proteins that shuttle between the cytosol and the nucleus, or a lot of proteins that are found in different components of the secretory pathway, for example, but also unexpected combinations, such as many proteins that are in organelles that are not in physical contact, proteins that are in the nucleus and the Golgi apparatus, or proteins that are in the nucleus and on the plasma membrane. So I think that there's a lot of knowledge that we can understand about the crosstalk between organelles and by looking at the multilocalizing proteins. And I also believe that multilocalizing proteins are interesting because they may have a context-specific function and even moonlight in different parts of the cell. And if this is true, it would increase the functionality of the proteome quite a lot, and also the complexity of the cell from a systems perspective. So I think that this is something we need to take further, consider more in systems biology studies, that proteins may be found in many places. Other images that we're interested in are images that looks like this. So this protein, this is a cell line that is, it's a regular cell line, non-synchronized cells, but this protein is only expressed in 2 cells out of these 15 cells. We also see proteins that are expressed in all cells, but at a low level and then maybe at a 100-fold higher level in other cells. And we also see proteins that change location within a population of cells. So this protein is found in the Golgi apparatus in most cells, but in these 2 cells that just divided, it's in the endoplasmic reticulum. So this led us to the hypothesis that maybe a lot of these variations are correlated to cell cycle progression, since these cells are not synchronized. And before I get into the details of the cell cycle progression, we can see that 16% of all proteins show such pronounced single cell variation, and I still think this is an underestimate. And we can also, the majority of these variations we can reproduce in all the cell lines that we're testing, so they seem to be pretty general. So we set out to determine which of these variations are correlated to cell cycle progression. And to start out with, we identified all the proteins that are spatiotemporally restricted to only be found in a mitotic cellular structure. And we could find about 160 proteins, of which 118 were previously not known to be in these structures, and it includes proteins in cytokinetic bridge or the mid-body or even the mid-body ring. So it gives quite detailed clues about which biological functions these proteins could be involved in. I should also say that this is yet unpublished work. And then we moved on to try to understand if the rest of the variations are correlated to cell cycle interface progression. And unfortunately, we're limited to static cells with the antibodies. So we decided to use the Fucci cell model to be able to resolve the cell cycle. So it's a cell model that is red in G1, and then the cells turn yellow in the transition to S, and then the cells are green. So we can make these plots, and this is red fluorescence, green fluorescence, so we can see that you can follow the trajectory of the cell cycle here by measuring the red and the green fluorescence. This is G1 cells, these are cells that are transitioning to S, and this is SG2 cells. So we took this cell line and then we stained it with the antibodies and with the aim to determine if the variations are correlated to interface progression. So if you look at this image in the middle here, it's DUSP18. You can see variation, and you can also see here that in the beginning of the cell cycle, there's low expression. Yellow means low expression, and then it gradually increases, red and blue. Blue is the highest expression. And if we look at these binned populations, we can also see that there's a significant difference. So this protein is increased over the course of the interface. Whereas the protein at top here, FAM71F1, we have dark and bright cells here in this image, and we can see that we have both high and low expressing cells in all phases of the cell cycle, as you can see in this plot, and there's no significant difference. So there's proteins where the variation is not correlated to the cell cycle, and we can identify the proteins where the variation is correlated to cell cycle progression. And then we can also fit a model to this cell cycle trajectory and actually make a plot over what the protein expression looks like over the pseudotime of this cell cycle. This is again DUSP18. DUSP is a member of the dual specificity phosphatase family of proteins, and a lot of them are known to be regulated by the cell cycle, but this one had no information like that, so we could show that it is regulated over the cell cycle, and it peaks in the late SG2 part of the cell cycle. What is interesting is also, since we have single-cell data, we can actually calculate the fraction of variance that is explained by the cell cycle. And I'm not going to show you more examples now, but it's interesting to see that some proteins, like DUSP18, a lot of the variation is explained by the cell cycle, but for other proteins, only part of the variation is explained by the cell cycle, but it's still significant. So in total, we were able to identify 400 proteins that are correlated to interface progression, and among these proteins, we have, of course, many known cell cycle proteins, but also many new proteins. So about 70% of these proteins have no known connection to the cell cycle if we look at gene ontology or proliferation. If we look at gene ontology, reactome, or phenotypes in cycle base. And if we look at studies of temporal gene expression at the transcriptome level, it's also very poor overlap. So we believe that a lot of these variations are maintained at a post-translational level, or maybe a translational level. And then if we turn to protein-protein interaction networks, this is a string plot, we can see that the known cell cycle proteins, a known cell cycle protein is here marked with a color, and the white nodes are proteins with no known connection to the cell cycle. We can see that they form this super tight cluster with interactions. But the novel proteins that we have identified, a lot of them are still not connected to this network, but we do see that they form this extended network and connects more loosely to this core. So we're very interested in understanding if it's just a consequence of the cell cycle progression, or if these proteins and these clusters actually contribute to the regulation of cell cycle too. And there's also many proteins of clinical interest in here, for example, some potential novel markers for cellular proliferation, but this is all yet unpublished and for another talk. So with that, I'm going to switch gears. Oh, one more thing I want to say. What is also interesting here is that we have the spatial resolution, so we can look at how proteins in different organelles are also coordinated by the cell cycle. We can see that some organelles tend to change more in some parts of the cell cycle, whereas other organelles tend to change later or earlier in the cell cycle. So there's a lot we can do here to try to understand cell cycling better. So to shift gears, everything that we do is critically dependent on the pattern classification in these images. So we have 1 million such images, and the better we can quantify the patterns that we see in these images, the better we can understand biology. And I'm going to talk to you about 2 different approaches we've used to classify these images. We've, of course, done it manually, but we've also done it with citizen science and with machine learning. But before we move on, I just want to remind you that this is indeed a challenging task for many reasons. There's a great class imbalance. We have common classes like the cytosol and rare classes where there's only 5 proteins in microtubule ends, for example. And there's also great morphological differences. We're working with a whole panel of different cell lines. Then we have the single cell variations and the multilocalizing proteins. So it's a very complex task. So we thought that there's a few good machine learning models there, but all of them are focused on one cell line and mainly single patterns. So we thought that maybe humans are really good at recognizing patterns. Maybe a citizen science approach would be more suitable. So we teamed up with MMOS, Swiss startup company that had the idea that what if we make a game? And there's a lot of nice gamified citizen science projects. But the idea here is that instead of making our own game, what if we can inject our scientific question into an existing computer game with millions of players already or something like that? So we drafted this idea about a minigame for pattern recognition in microscope images, and we teamed up with CCP Games, which are the producers of EVE Online, a massively multiplayer online science fiction game. And we created something called Project Discovery. And I'm now going to show you a movie that was broadcasted when this minigame was released in the universe of EVE Online in March 2016. And this is a news broadcast that was broadcasted in-game, so that you get a feeling for what we're trying to do.
The sudden drifter pullback to the 5 known hive systems are believed to be connected to the Capsuleer reports that gate restrictions in those systems have been removed, clearing the path for Capsuleers committed to pushing back the drifter menace. The conflict has certainly taken its toll on the Amar military, and talks of the withdrawal being a reaction to Sisters of Eve, and Capsuleer actions only adds insult to injury for the Amar. Meanwhile, Sisters of Eve has launched a crowdsourced analysis of collected drifter data, entitled Project Discovery, and rallied under the banner of Citizen Science Begins With You. They hope to unlock the new drifter tech so that they may be made public and shared with all. This is Lena Amber, reporting for The Scope.
Yes, it is. You're paying attention. Nice. I'll show you. So what I wanted to show you there is that we wanted to make a seamless gaming experience. So this demonstrates how the game designers try to interweave the scientific story into the story of this virtual universe. So in this universe, there's these creatures called the drifters. They are invading, they're scary, they're powerful. So then a more friendly association called the Sisters of Eve are collecting drifter data, which is the images. And then they launched a crowdsourced effort in-game, led by Professor Lundberg. In-game. And they're asking the players, help us to map these drifter images. And if you do that, you can actually maybe understand the drifters and clone their tech. That was the pitch. The game in itself was quite simple. I don't have any time to go into any details. The gamers see an image, they go through a training, a tutorial, they see an image, they have reference patterns. We're asking them, which patterns do you see in these images? There's also levels and badges and swag and everything to drive motivation in-game. I'm still waiting for the real-life combat suit. I haven't gotten it. So it turns out that we ran this project for a year, and it exceeded all our expectations. So during 1 year, we had 322,000 players that helped us classify our images, which is more than any other citizen science effort. So tapping into computer games is quite efficient way of reaching out to people. They provided 32 million image classifications, which is mind-blowing. And if we look at the time they spent on the minigame, they spent 70 working years, which is crazy. So after we closed this game, after 1 year, we spent another year analyzing this data. And I unfortunately don't have any time to go into the details. I'm just going to summarize the results in one slide. And in parallel to this, we also developed machine learning algorithms to classify mixed patterns across multiple cell lines, which has never been published before either. And it turns out that gamers and machine learner, they perform equally, good or bad, however you see it. Macro F1 around 0.5, which is still better than anything published. But what is interesting is that the gamers perform better than the machine learner when it comes to rare classes, and they can even discover new things. So we had an option, like if you see something weird, let us know, and they can discover new things. We could also see that the machine learning and the gamer, they're good at different things. So if we make a model that mimics the gamers and augment our model with that, the new model actually outperforms both models. And we also blindly tested experts, and experts still outperform both, unfortunately. But this model is still good enough for us to use in the Protein Atlas, and we can skip a lot of manual annotation as a first round. So this project has been very fun, and it also contributed a lot, actually, both in terms of novel biology to the Protein Atlas. We've been able to find about 10 new rods and rings proteins, in addition to the 3 known ones. We've taken our database from 23 to 30 locations, and we also published this last month in Nature Biotech, and we weren't able to honor the gamers with, I tried to include them as a consortia, as authors, but we didn't do that, but we got a spaceship on the cover at least, so that's something. So if you're interested in the machine learning and the idea of classifying patterns, please read the full paper or talk to me later in a break. But to summarize it, I think that we demonstrated a path forward to how massively multiplayer online citizen science can feed into tailored machine learning algorithms for scalable and improved data classification. And I also think that tapping into computer games as a portal for harnessing the brain processing powers of humans is something that we'll see a lot more of, both in science and in healthcare. And with that, I would like to thank my funders, the amazing team that made all of this possible, and in particular for this talk, Diana Medesian, Devin Sullivan, and Casper Vinsnes. And then before, I would just like to leave you with this. If you're interested in classifying patterns in human cells, we're now hosting a Kaggle challenge. It's active right now, where we want you to beat the experts, beat the gamers, beat our machine learning model, but develop a tool that is better than the experts. That would be amazing. And you have the chance of winning some nice money and being co-authors of a paper and so on. So please participate and help us spread the word. Thank you. And I guess I will. I'm actually sharing this session, so I'll ask you if you have any questions before I move on to the next speaker. Yes, go ahead.
All right, so with the game, if your experts aren't like sort of authoritative, if your experts aren't authoritative and being able to classify these things, and you know your gamers are kind of bad at it, how do you actually get a gold standard of saying, you know, my gamers are 50% F1 score?
That's a very good question. So for the gold standard, we use the data that we have in the Protein Atlas, and that's images that are annotated by 3 different individuals, and then we look at many images from the same sample, and then we look at many images from the same sample replicates in different cell line, and then we sometimes also augment it with other known information about that protein. So that would be the gold truth. Whereas the experts that we tested, that's one expert at a time, seeing only one image at a time.
Thank you.
Yes, over there.
So I want to go back to the first part of your talk. So you mentioned that a lot of proteins, information about their location, et cetera, was not known. And I'm wondering, what I wanted to know is, how many of those proteins were annotated against model systems? And so were they not known anywhere, or were they just not known for humans?
Some of them were known, of course, in mouse, for example, and in yeast and so on. So some of them were known. The majority were still unknown. Some of them also had bioinformatics predictions of where they would be, that they have a signal peptide or some predictions like that. So for some of them, there were clues, but no experimental data. But the majority were still unknown. It's hard because UniProt don't version their databases. That's actually a problem for us. Yeah, this is one last question.
That was great. I was wondering, for the proteins that showed varied expression but was not dependent on the cell cycle, did you then also classify them into organelles that are short-lived, for example, early endosomes that progress into late endosomes, so that way you can tell why the expression is varying?
That's a very good question. So, in fact, the majority of the proteins that show single-cell variations are not correlated to the cell cycle. We have 800 proteins, and we can see that these are mainly located to different organelles. The endosomes are not the major organelle, but we see a lot of mitochondrial proteins, transporters in mitochondria, proteins in intermediate filaments, but also nuclear and cytosolic proteins. And we're still trying to understand the function. We're trying to correlate it to everything we can to understand if it's some kind of bed hedging strategy or if it's determined by other deterministic factors. We don't know, but any ideas here we're welcome to take, but that's for another talk. So thanks a lot for your questions and listening to me. And with that, I would like to introduce the next speaker, Nozomu Yashi from the University of Tokyo. Is going to talk about chasing molecular and cellular dynamics using DNA barcodes.